Good evening, everyone. My name is Dora Markati and I am a researcher at the University of Oxford. I'm a member of Florence team in the Department of Pediatrics, and together with our group, we are organizing the Natural History Study, which is supported by FAST UK. Tonight, I will talk to you about the current landscape of Angelman Syndrome therapeutics. I will analyze both the preclinical and clinical developments, as I believe that it is important for everyone to have the biggest picture possible of not only what is currently going on, but also about what is going to happen in the near future. Before analyzing the different therapeutic categories, I would briefly like to give you a simple outline of Angelman syndrome pathology. I believe that this is very important because it will help us understand the different levels at which these different therapeutic categories or different therapeutic weapons can act on. Now, before explaining this, I would also like to refer to a very basic biological process which will help us understand better the information which follows. Now, our body consists of proteins. The information for the production of these proteins lies in specific sequences in our genetic material, in our DNA. These sequences are called genes, and for every gene that we have two copies, which we receive one from our biological mothers and one from our biological fathers. Now, to start with, Angelman syndrome is caused by deficiency of the UBE3A protein. The UBE3A protein is a protein that participates in the recycling of other proteins in the nervous system. Consequently, deficiency or abnormal function of the UBE3A protein leads to increased or decreased levels of other proteins in the nervous system. Now, the cause for the UBE3A deficiency goes down to the level of genetics, to the level of our genes. The cause lies in the maternal copy of the gene, where this specific coding sequence that has the information for the production of the protein is either missing or is disrupted. The paternal copy could compensate for this, but in the specific case of the UBE3A gene, there is a naturally occurring long molecule which blocks the paternal copy. In other cases, some Angelman syndrome patients receive two paternal copies of the gene which are both blocked by this long molecule. And at this level, at the level of genes, is exactly where the first big therapeutic category acts on. The first therapeutic category, which could also be referred to as upstream treatments, includes mostly genetic therapies which aim to restore the UBE3A in the brain cells of Angelman syndrome individuals. To continue with the pathology, the UBE3A deficiency and the disrupted recycling of proteins in the brain lead to disruption of communication between the neurons. As you might have heard already, processes like the synaptic plasticity get impaired. Roughly, synaptic plasticity is the way our brain cells connect, but also the way the existing connections adjust, subject to different inputs which we receive either during development or at adult stages. And this is very important for specific functions to emerge. Now, the disruption of such physiological processes is much more complicated, and if we were to create a diagram of all these different biological processes, it would look like a map of different pathways, most of which we are still exploring. Each pathway might be responsible for one or more symptoms, and some molecules are key to these pathways. Now, these molecules are the targets of the second big therapeutic category, which is also referred to as downstream treatments. Downstream treatments could also be called symptomatic treatments because they aim to treat one or more symptoms that we see usually with individuals affected by Angelman syndrome. I will start by talking about the upstream treatment. The first subcategory of upstream treatments is the gene therapy or gene replacement therapy. Now, what happens with gene therapy is that we use viruses as vectors to deliver in the brain cells of individuals affected by Angelman syndrome a normal copy of the UBE3A gene. These viruses are harmless, and that is because in the lab we have replaced the sequences of their genetic material that make them pathogenic by those sequences which are useful in our case. Practically, what happens is that several viruses get modified in the lab in order to receive and to carry a normal UBE3A copy. Then these viruses are injected treated in the patient's spinal fluid, and they find their way to the brain cells. Then these viruses, which carry the normal gene, infect in a positive way the brain cells with this gene copy, and this becomes part of the genetic material of the cell. The cell recognizes this UBE3A gene that cells received from the virus as part of the genetic material, and they follow the processes that lead to the production of the UBE3A protein. Now, the proof of concept that something like this can work for Angelman syndrome came from preclinical studies which used Angelman syndrome mouse models. Here, I have the proof of concept paper which was published in 2011, and here is a very characteristic figure from the paper where you can see in brown the cells which produce the UBE3A protein. In the right column, the upper picture shows in brown color, as I said, the cells that produce the UBE3A protein, and as you can see, the levels of brown color are much more compared to the picture just under, which shows an Angelman syndrome mouse model. Now, the big picture on the left column shows an Angelman syndrome mouse model after treatment, and as you can see, the levels of brown color, the levels of cells producing UBE3A protein, are similar to those of the neurotypical mouse in the first picture. Now, the way by which we deliver these viruses into the spinal fluid of the patients is usually a process called intrathecal injection. This process is nothing different to a lumbar puncture. Lumbar puncture is a process which is very often used in clinical practice, also for diagnostic purposes. Now, there is another type of gene therapy treatment which is slightly different methodologically. This is called cell therapy. In this case, patients' own blood cells are collected from a vein and they are infected with a normal copy of the gene outside of patient's body in the lab, again using a viral vector. Then, the successfully infected cells are infused back to the body from the vein and find their way to the bone marrow as they are programmed to do so. Then they mature to cell forms that cross the blood-brain barrier and get into the nervous system. Now, these cells, because they have a normal copy of the UBE3A gene, they produce the UBE3A protein. And then they give this protein to the brain cells which are deficient in a process called cross-correction. Now, at this point, I would like to clarify that gene therapy is a therapeutic category under which there are several different programs led by different institutions and different companies. We still do not know which one of these programs will have the best profile of efficacy or the best profile of safety and for which genotypes. This is why it's really exciting and really crucial that we have so many developments going on. Overall, this therapeutic category is very close to human clinical trials, but not yet started. Now it's time to move to the next big category of treatments. This category, which is also a subcategory of the upstream treatments, includes all these treatments that aim to unsilence the paternal copy. As I said before, the paternal copy is silenced by a naturally occurring lock molecule which selectively blocks the sequence of the paternal gene from revealing the information required for the production of the UBE3A protein. Now, this long molecule is called Antisense transcript. So, again, this second category of treatments interacts with the existing genetic material of the patient and aims to unsilence the paternal copy by stopping this long molecule, the antisense transcript. One way of doing this, which have been proven very efficacious in animal studies, are the antisense oligonucleotides. the antisense oligonucleotides are sequences which are complementary to those to this antisense transcript. When we administer antisense oligonucleotides in the spinal fluid of patients, again by using the same process that I described before, these antisense oligonucleotides find their way to the brain cells and they hybridize with the antisense transcript. Now, this hybridization leads to the degradation of the antisense transcript, and that leaves space for the paternal copy to give the information to the cell for the production of the UB3A protein. Now, as with gene therapies, there are several programs of antisense oligonucleotides currently in development. The great news is that this therapeutic category is currently being assessed in clinical trials. And we have two therapeutic programs currently in clinical trials. The first one is the antisense oligonucleotide produced by Gentex Ultragenyx, which is currently in phase 1-2. The patients who can be included in this clinical trial are those patients that have a maternal deletion and are between 4 and 17 years. Now, the way of administering this antisense oligonucleotide is again, as I said, by intrathecal injection, or the process called lumbar puncture. The locations at which this clinical trial is taking place are the United States, Canada, and the United Kingdom. The second program, which is currently at clinical testing, is the antisense oligonucleotide produced by Hoffmann-La Roche. This is in phase 1. And again, patients who can be included in this clinical trial are patients who have a maternal mutation or deletion less than 7 megabases. The age of the patients who can be included is from 1 to 12 years. Again, the way of administration is a lumbar puncture and intrathecal injection, and the locations at which this clinical trial is taking place are the United States, the Netherlands, Italy, and Spain. Now, except for the antisense oligonucleotides, there are another 3 therapeutic categories which aim to unsilence the paternal copy of the UB3A gene. These include the topoisomerase inhibitors, the CRISPR-Cas tool, or otherwise known as the genetic scissors, and the zinc fingers. All of these have already shown efficacy in preclinical studies, like studies that use Angelman Syndrome animal models. And to be more specific, these animal models were mice models in this case. So here I have created a snapshot of how the development of the different upstream therapeutic approaches looks like currently. Here you can see all the information about the different programs in development and the overall phase of development per therapeutic category. Overall, as you can see, there are 9 different therapeutic approaches of upstream treatments, and 3 of them are already in or close to clinical testing. And this is a really impressive picture because, specifically talking about Angelman Syndrome, because it is a rare disease. Now, additionally to those that we have already discussed, and you can see here in this diagram, there are another 3 therapeutic approaches which are still at discovery level. This means that we believe that they are promising, but they have not been tried yet on animal models or in human studies. These include the enzyme replacement therapy and therapies using short hairpin RNAs or microRNAs. I will now talk about the downstream treatments. These treatments that could also be called as symptomatic treatments. So here is a snapshot of how the therapeutic pipeline of downstream treatments this time looks. As you can see, there are another 6 different therapeutic approaches in this category of downstream treatments. Now, most of the downstream treatments are aiming to improve the function of the brain cells or to improve processes as the synaptic plasticity that we discussed earlier. Other downstream treatments aim to treat specific symptoms like the non-epileptic myoclonus or epilepsy. Some of these downstream treatments might be applicable to other diseases as well. Other diseases might not have exactly the same cause as Angelman syndrome, but might have same consequences on brain cells, and therefore they might present with symptoms similar to Angelman syndrome, and that's why they can benefit from the same therapeutic approaches. Now, downstream treatments cannot cure Angelman syndrome. However, there are 3 positive things about downstream treatments. First of all, they are already in clinical testing, and their safety profile has been or is being assessed, at least for most of them, in healthy individuals or in individuals affected by other diseases. This means that we have already a very good picture of their safety and tolerability profile. Secondly, they are applicable to all different genotypes of Angelman syndrome. And last but not least, they can be combined with the genetic therapies that we discussed before, and they can enhance the overall effect of the treatment scheme. Recently, in December 2020, the development of a compound with the name gaboxadol, which has progressed as far as in phase 3 of clinical development, has been seized for Angelman syndrome, and that was because the compound failed to show efficacy. Now, the main measure of efficacy, or otherwise primary endpoint as we refer to in the clinical testing of gaboxadol, was a 7-point rating scale, which was assessing patients' improvement on the compound compared to the baseline before treatment was given. And that assessment was for 5 key areas, 5 key domains which are usually affected by Angelman syndrome. Now, gaboxadol was the only compound that has progressed that far in clinical trials for Angelman syndrome, and we've been taught a very important lesson. In order to assess the efficacy of all these different treatments that we discussed so far, we have to have in our hands the appropriate, the suitable assessment tools that will help us lead to safe conclusions. At the moment, the existing developments that I have just presented to you bring us in front of one need, and this is the need of having objective and cross-comparable measures in our clinical trials. Now, in order to develop these meaningful outcome measures for the different domains affected by Angelman syndrome, we need to work all together, both researchers and families, in prospective studies, and we need to do that in parallel with the clinical trials. So the start of the clinical trials does not exclude the need of having studies of such type, which are like natural history studies or biomarker studies. And talking about biomarkers, we also need to design and develop digital or lab-based biomarkers, because these are the markers that will help us, that will inform us objectively about the progress of a patient on treatment. Now, only by having tools like these in our hands will we be in a position to design better and more efficacious clinical trials. And by better and efficacious, I mean clinical trials which can inform us safely about the efficacy of a treatment in a smaller amount of time and in a smaller population of patients. And this is particularly important for a rare disease like Angelman syndrome. Now, these tools are also the tools which we will use when we want to prove the efficacy of a treatment to the different regulatory authorities when we believe that the treatment works. And these are the same exact tools that will help us get compensation by the different health systems for all our patients when a treatment is approved and is becoming standard of care. Last but not least, I would like to briefly take you through some key messages. These messages also answer questions which we have received from you before this webinar. Overall, a total of 15 different therapeutic approaches is currently in development for Angelman syndrome. Now, different drugs might benefit different genotypes. We still do not know which of the different therapies will help which of the different Angelman syndrome genotypes and in what combination, or even in what chronological order. Mostly referring to genetic therapies, these are still at early stages of clinical development. Now, this means that these stages of clinical development aim mostly to assess safety and tolerability and to a lesser extent efficacy. We cannot yet make safe conclusions about the efficacy. This will need trials with larger population and bigger duration in order to answer the question, does the treatment work? Now, the different genetic therapies have different advantages and disadvantages. For example, gene therapies are considered one-off treatments, whereas antisense oligonucleotides require dosing. But from the other side, gene therapies are very likely to induce the immune reaction of the treated individual against the virus. Now, conclusions about the overall safety and efficacy profile of the different treatments require years of clinical research. As I mentioned before, the combination of different genetic treatments or the combination of downstream with genetic treatments might potentiate the effect. At the moment, all we can do, and the best we have to do, is participate in natural history studies and biomarker studies because these are equally crucial to clinical trials. This will help us collect the keys to unlock all the previous questions with regards to safety and efficacy. The therapeutic pipeline for Angelman syndrome looks very complicated, but this is a good thing, because this means that several researchers, several institutions and several companies work together, united, in the effort to treat Angelman syndrome. But because it's complicated, we have gathered all the information in a scientific paper, which you can find here on this slide. The name of it is Therapies in Preclinical and Clinical Development for Angelman Syndrome, and this has been published already in the journal Expert Opinion on Investigational Drugs. It is an open access paper and has information about results from preclinical research and tables which will help you get a good grasp of the current developments both at preclinical and clinical setting. Now, if you wish to discuss more or to participate in the natural history study or the clinical trial which will start in Oxford, I would be more than happy to receive your emails. You can find my contact details here on this slide, as well as online at the official website of the University of Oxford. Thank you so much for your attention tonight. Good night.